Mitosis is the process of cell division, where a single cell divides. It results two identical cells with the same number of chromosomes. My teacher told me at school what mitosis process was. She told me it has 4 phases, starting with prophase. During this material, it starts to condense. After that, the chromosome begin to become visible. Mitosis process has 4 phases too, but they start with P, M, A, T, and are easy to find. They are quickly to learn and easy to understand. They start with P, M, A, T, and are easy to get. Ooh, there ain't no other way. It is easy to understand. Prophase is the first. Ooh, there ain't no other way. It is easy to understand. Prophase is the first. Start with P, continue M. Next is A, finally T. Start with P, continue M. Next is A, finally T. Then we go to anaphase, where chromosomes separate. They move along spindle fibers to the end of the cell. Then it comes finally telophase, where distinct chromosomes separate. Mitosis process has 4 phases to pass. They start with P, M, A, T, and are easy to find. They are quickly to learn and easy to understand. They start with P, M, A, T, and are easy to get. Ooh, there ain't no other way. It is easy to understand. Prophase is the first. Ooh, there ain't no other way. It is easy to understand. Prophase is the first. Then there is meiosis, the process of dividing. It has 2 ways of doing that, meiosis 2 and meiosis 1. In prophase 1, cousin overpass. In metaphase 1, spindle fibers attach. On telophase 1, spindle fibers dissolve, where 2 haplocells are formed. Oh, we missed anaphase. It comes before telophase, where chromosomes of the ends are pulled out. Meiosis 2 or meiosis 1, it doesn't matter which one. They are both easy to understand and fast to learn them. Meiosis 2 has the same rules, starting with 2 haplocells and ending with 4. It's easy to get this long process. Just practice the M. Ooh, there ain't no other way. It is easy to understand. Prophase is the first. Ooh, there ain't no other way. It is easy to understand. Prophase is the first. This was mitosis, hey. This was meiosis, hey. Start with P, M, A, T, and finally T. It's easy to understand. It's with P, M, T, and that's how this song finally ends.
What is mitosis? Mitosis is a process where a single cell divides into 2 identical daughter cells. This is also known as cell division. During mitosis, one cell divides once to form 2 identical cells. The major purpose of mitosis is for growth and to replace worn-out cells. If not corrected in time, mistakes made during mitosis can result in changes in the DNA that can potentially lead to genetic disorders. In the process of mitosis, we have 5 different phases. The first one is interphase. In this phase, the DNA in the cell is copied in preparation for cell division, and this results in 2 identical full sets of chromosomes. The second phase is prophase. In this phase, The chromosomes condense into egg-shaped structures that can be easily seen under a microscope. At the end of prophase, the membrane around the nucleus in the cell dissolves away, releasing the chromosomes, so the chromosomes can replicate. Then we have the third phase, which is metaphase. In this phase, the chromosomes align at the metaphase plate, and the mitotic spindle fibers attach to each of the sister chromatids. Then we have the anaphase. This is a simple phase because in this phase we just have that the sister chromatids are then pulled apart by the mitotic spindle, which pulls one chromatid to one pole and the other chromatid to the opposite pole. Then we have the telophase and the cytokinesis. At each pole of the cell, a full set of chromosomes gather together, and a membrane forms around each set of chromosomes to create two nuclei. The single cell then pinches in the middle to form two separated daughter cells, and each containing a full set of chromosomes within a nucleus. This process is known as cytokinesis.
Meiosis is a process in which the number of chromosomes per cell is cut in half through the separation of homologous chromosomes in diploid cells. Meiosis usually involves two distinct divisions called meiosis 1 and meiosis 2. Let's talk about meiosis 1. Meiosis 1 has the cells go through phases to make the separation. These are four phases: is interphase, prophase, metaphase, and anaphase, telophase, and cytokinesis. The interphase is when the DNA is copied, resulting in two identical full set of chromosomes. Then they pass to the prophase, in which prophase of meiosis each replicate chromosomes paired with the corresponding homologous chromosomes. They're pairing from a structure called tetrad. Then it passes to the metaphase. During the metaphase of meiosis, paired homologous chromosomes line up across the center of the cell, and then it goes to anaphase. The anaphase spindle fibers pull each other's homologous chromosomes paired toward opposite ends of the cell. Then it passes to telophase. The telophase, in which the nuclear membrane forms around each cluster of chromosomes. Then the cytokinesis, forming two new cells. Then we pass to the mitosis 2. The mitosis 2, the two cells now enter second meiotic division. Like the first division, neither cells goes through the round of chromosome replication before entering meiosis. They have the same four phases: prophase, metaphase, anaphase, and telophase, and cytokinesis. In prophase 2, at the cell enters prophase 2, their chromosomes each consist of two chromatids. The chromosomes do not pair from the tetrad because homologous pairs were already separated in meiosis 1. Metaphase, anaphase, and telophase and cytokinesis mostly do the same as meiosis 1. The metaphase do pair homologous chromosomes line up across the center of the cell. Anaphase does the fibers pull each other's homologous chromosomes toward opposite ends of the cell. Then the telophase, in which makes the nuclear membrane forms around the cluster of chromosomes. And the cytokinesis makes now four cells, because in this meiosis enter two cells. Now we'll be making four new daughter cells.